The Southern blot was named for its inventor, Edwin Southern, and the names of the other types of blots we'll talk about, Northern and Western, are kind of playing off this name. The purpose of a Southern blot is to detect specific sequences of DNA, which can help you diagnose genetic disorders or the presence of particular viruses or bacteria, or even be used for forensic applications, such as whether a drop of blood came from a certain person. First, you have to load your DNA of interest into a well at this end of the gel. Next, you apply an electric current to the gel with a negative charge at the top end and a positive charge at the far end. Do you know why you align the charges this way? It's because DNA is negatively charged, so it will run away from the negative end towards the positive charge. Since the gel is basically a tightly woven sponge of fibers, larger pieces of DNA take a long time to get through the gel, whereas small pieces get through pretty quickly. This principle is what allows you to separate strands of DNA by size. When you've been running this long enough that the smallest strands of DNA are nearing the end of the gel, you shut off the electric current and transfer the DNA to a filter, where the DNA is more stable and can't diffuse around. At this point, you can't see what's on the filter, but you can use a radio-labeled DNA probe that is complementary to, and therefore will stick to, whatever sequence you're interested in. So for example, let's say you suspect your patient has a particular viral infection, and you know that virus has a certain sequence of DNA that is not found in humans. You can create an oligonucleotide that is complementary to that sequence and attach a radioactive label to it so you can see it later, and then wash it over your filter. After you give it some time to stick to its target, you wash it away, so the only probe left is in the location of its complementary target sequence. If the target sequence that you're looking for was not in your original sample, then the labeled probe will all wash off and you won't see anything. But if it was in your sample, then it will stick to its complementary sequence and will show up as a band on the filter on the X-ray film. This is the only thing you'll see, since only your probe is radio-labeled, so the rest of the DNA on the filter won't be visible. A Northern blot is basically the same thing, except now instead of running DNA through your gel, you're looking at RNA. So this can be used to measure the quantity of RNA, or just whether a certain sequence of RNA is present. This is often used to determine how much a certain gene is being transcribed into mRNA. A Western blot also uses a similar concept, except now you're looking at proteins. Since proteins can't be targeted by using complementary strands, you instead have to use labeled antibodies, which are specific to your protein of interest. One example of when Western blots are used is to confirm the diagnosis of HIV after it has been detected using an ELISA, which we'll discuss shortly. Western blots can be used in any case when you want to know how much of a certain protein you have in your sample. And lastly, a Southwestern blot is kind of a hybrid between the Southern and Western blot, so you can use its name to remember this. The goal here is to detect proteins that can bind to a specific sequence of DNA, such as transcription factors and nucleases and histones. The way you do this is to run a protein through a gel just like you would during a Western blot, but then use labeled oligonucleotides as probes instead of antibodies. If the DNA probes stick to a protein, you will see a signal at the location in the gel which corresponds to proteins of a certain molecular weight, and that lets you know which proteins can bind to that sequence of DNA. Let's quickly review with a flash quiz. A Southern blot is used to visualize blank, a Northern blot is for blank, and a Western blot is for blank. So this would be DNA, RNA, and protein. By the way, I like to remember this using the top half of a picture of a cell. So here's our nucleus at the bottom, and here's our plasma membrane at the top. The DNA is in the nucleus, so this is the southern region for Southern blot, and then it gets transcribed, and the RNA is in the north for Northern blot, and the RNA gets translated, which I'm going to draw in the west, so the protein is in the west for Western blot. If you don't like this, then don't use it, but hopefully it helps some people.